Hello everyone. In today's presentation, we'll have a look at a comprehensive presentation on the nitrosamine impurities. So we'll look at the major events of the impurities right from their discovery to the other events that led to the analysis and the limits determination. We'll have a look at the USFDA guidance for the control of nitrosamine impurities, which was initially published in September 20 and later revised in February 2021. And we'll have a look on the latest nitrosamine drug substance related impurities limit guidance that is issued in August 2023. Talking about the nitrosamine impurities, by this time we all know that nitrosamines are the class of compounds having chemical structure of a nitroso group bonded to an amine. Okay, they can be formed by the nitrosinic reaction between amines, either secondary, tertiary or quaternary, and nitrous acid, that is salts under acidic condition. So in the USFDA guidance, there are specifically seven nitrosamine impurities defined: NDMA, NDEA, NMPA, NDIPA, NIPEA, NDBA, and NMBA, right? Apart from these seven nitrosamine impurities, now after the development studies, there are other compounds described as nitrosamine drug substance related impurities. Okay, so what are these? They share the structural similarity to the API, having API or API fragment into their chemical structure, and are unique to each API. They are formed in the drug products through nitrization of APIs or the fragment of APIs that have secondary or tertiary amines. And they lack the carcinogenicity and mutagenicity data derived from the animal studies. So why is there so much of chaos around nitrosamine impurities? Because they are potential genotoxic impurities and are referred to as cohort of concern in ICH M7, right? They may increase the risk of cancer if people are exposed over a long period of time. So when we talk about the discovery of these, there are specifically three types of medicines that led to the discovery of these nitrosamine impurities. The first one in June 2018 were the blood pressure medicine. The second one in September 29 were the acidity medicine, and the third in December 2019 was the diabetes medicine, right? So the blood pressure medicine that was discovered for the nitrosamine impurities in June 2018 were angiotensin 2 receptor blockers, specifically the sartan families. Example, valsartan, irbesartan, losartan, etc. In later, during the analysis and the studies, the acidity medicine discovered nitrosamine impurities in September 19, that is histamine H2 receptor blockers, which are ranitidine and nizatidine specifically. And thirdly, the diabetes medicine, that is the regulation of glucose and insulin medicine, which are metformin. So impurities were discovered specifically in the metformin extended release tablets. It should be noted that though the discovery was limited to these three categories of medicine, the current guidance and the recommendation apply to all the chemically synthesized APIs, drug products containing chemically synthesized APIs, and whatever drug products are at risk. Before moving ahead, please ensure that you have subscribed to the channel Prof Talks and have pressed the bell icon so that you can receive the notification of upcoming presentations. We'll look into the timeline of the sartans that discovered the nitrosamine impurities, right? So in June 2018, an organization in US, that is Princeton Pharmaceuticals, reported that they have NDMA impurity in valsartan, which was sourced from a Chinese company, Zhenjiang Huai Pharmaceutical, referred to as ZHP. So FDA moved into an action mode and identified four other manufacturers which were using the same API from ZHP. Also, they extended the investigation to other valsartan API manufacturers. By July 2018, several of the organizations started recalling the medicine valsartan due to the API of ZHP that contained the NMDA impurity, right? And according to ICH M7, a specific limit of 96 nanograms per day was determined to be as safe. In August 2018, USFDA released a GCMS method for quantitation of NDMA in valsartan API and the product, and mentioned that it should be validated before use. In September 2018, another impurity, that is NDEA, was determined in valsartan from same API manufacturer ZHP, and which led to an import alert to the ZHP organization by USFDA. In October 2018, USFDA published the GCMS method for the NDEA impurities. Later, there was recall of irbesartan due to NDA impurities. Then there was recall of losartan and valsartan, and the USFDA issued warning letter, right, to ZHP. In February 2019, USFDA provided the limits of all the nitrosamine impurities based on the specific calculations which were considered to be safe. So this values can be referred to from the current guideline, also from the current guidance also, and these are the acceptable limits that are defined in milligram per day and nanogram per day for some of them, and ppm as well, right? And in June 20 there was import alert for multiple organizations related to this type of impurities. Coming to the ranitidine story, the timeline for the ranitidine medicines. So in September 2019, there was a first publication that the FDA is evaluating the risk due to presence of low level of ranitidine, low level of impurities in ranitidine, which led to substantially voluntary recalls of ranitidine by some firms in September 2019. And in October 2019, USFDA issued a statement that GCMS method is which was issued for the sartan family may not be specific for the ranitidine because it leads to the heating of sample which might increase the nitrosamine impurities, right? And there was an announcement in December 2019 to test all lots, and in substantially April 20 they requested to stop all the ranitidine medicines. The metformin timeline: in December 2019, a statement was issued that there are recalls outside US for NDMA impurity in metformin. However, the FDA is still investigating and there are no recalls as of now in the US. In February 20, the lab tests of metformin established a limit of 0.096 microgram for NDMA. No lot of metformin were found to have limits above this and no recalls were recommended. Then there was recall of metformin ER, reported high levels of NDMA in May 2020. So they were recalled by some firms like Apotex, Amneal, Teva, etc. In July 20, the FDA issued paper on the testing of metformin and established that there is a difference in results between testing by some external labs for metformin ER and the testing done by the FDA, and the method used by the external labs may be inappropriate. But still several of the organizations started recalling the metformin ER medicines, and the recall continued till June 2021. Specifically for the guidance of industries and the publications. So the first guidance for industry was on this nitrosamine impurity was published in September 2020, named as Control for Nitrosamine Impurities in Human Drugs, and it recommended a three-step process for the manufacturers to take to detect and prevent objectionable level of nitrosamines, right? In February 21, there was an update to this in which there was timeline provided for step 1, that is we'll look into the 3 steps in detail, of which the step 1 is the risk assessment. So the guidance provided a timeline of March 2021 for completion of the risk assessment. In November 21, there was an update which was not specifically a guidance but an update provided that timelines for confirmatory testing, that is step 2, and reporting changes to the product, step 3, was October 2023. And latest in August 23, they have published a guidance on nitrosamine drug substance related impurities. In the first slide itself, we have seen what are nitrosamine drug substance related impurities. So now the FDA has published a guidance for the recommended acceptable intake limits for the NDSRIs. Before going into the details of both of these guidance, that is the control for nitrosamine and the recommended limit for NDSRIs, if we can summarize what we have seen till now. So for the nitrosamine impurities that were detected and reported into the product, so FDA considered impact onto the products that were into the market, that they investigated the root cause for the occurrence, and control on the products before release into the market. So for impact on products in the market, they developed the analysis method, established the acceptable intake limit which should not be exceeded and which is considered to be as safe, tested all the products in the market to determine the level of impurities, recalled the impacted products which were having the impurities about the defined acceptable intake levels, investigating the root cause, the detailed investigation for identifying the root cause in coordination with various cross-functional teams from various pharmaceutical organizations. They identified the potential causes as the manufacturing process, the chemical structures of the API, storage or the packaging conditions, and the formation in body due to processing. And for control on the products before release, they established that manufacturers are responsible to control the presence and test the products for nitrosamine. They released the guidance for industry, which had all the details of the steps to be taken. The step 1 that we are specifically talking about is the risk assessment that need to be done for all the products, right? And the step 2 is whenever a risk is identified, a product is identified to be at risk on the step 1 risk assessment, a confirmatory testing has to be done for that product. And during that confirmatory testing, if the limits are identified to be as high, then there has to be changes to the manufacturing process to control the level of the nitrosamine impurities. So these are the 3 steps of which the guidance talks about. Moving to the first guidance, the control of nitrosamine impurities in the human drug, which was revised as revision 1 in February 2021. So it includes the introduction. In the background, it defines what are nitrosamine impurities, the general root causes, and nitrosamine impurities in drug products from other sources than API. And it gives the recommendation for the acceptable intake limits for the API manufacturers, for the drug product manufacturers, the details for maintaining the drug supply and reporting the changes to the FDA. We'll look into the details of all of this. So this guidance talks about 2 specific root causes for the presence of nitrosamine impurities, the presence of nitrosamine in the APIs and the nitrosamines in drug product from sources other than APIs. So the details for the nitrosamine in APIs, there are some general conditions in which the nitrosamines can be present. There are sources of secondary, tertiary and quaternary amines, contamination in the vendor sourced raw materials, recovered solvents, catalysts and reagents, the quenching process and the lack of process optimization and controls. And from other sources, they can be through excipients, they can be through the potable water and the product degradation to form nitrosamine impurities during storage. Going into the details of root cause for presence of nitrosamine in APIs. So the first thing was general conditions. So they are possible in presence of secondary, tertiary or quaternary amines in the nitrite salt under acidic reaction. Nitrite salt form nitrous acid which reacts with amine to form nitrosamine. So this is a greater risk when quenching process is used. Nitrites used as reagents in one step can be carried over to the next steps. And processes that use nitrites in the presence of secondary, tertiary or quaternary amines are at risks. The second thing was the sources of secondary, tertiary and primary amines. So may be present in the manufacturing process. The amines may be present in the manufacturing process. API may contain a functional group or they may be added intentionally as reagent and catalyst. So all of these amines can react with nitrous acid or other nitrosing agent to form nitrosamines. The amide solvent susceptible to degradation and amine impurity levels that may lead to nitrosamine contamination of API is process dependent and should be determined by each API manufacturer, right? The third thing is the contaminated vendor sourced RM. So if starting material and the raw materials are contaminated, like when fresh solvents are contaminated during shipment from vendors, sodium nitrite, potassium nitrite, etc. And each API manufacturer should determine the tolerated nitrite impurity limit. Cross contamination at the manufacturer when nitrosamine is produced in some other process, and awareness of supply chain is a must for RM for preventing the contamination. Continuing with the root cause for nitrosamine impurities in the APIs, recovered solvent, catalysts and reagent. If the recovery process involves a quenching step, nitrosamines may be entrained if they have boiling points or solubility properties similarly to the recovered materials. Some drug products using API manufactured by certain low risk processes, and firm may produce an API by more than one process that use common solvents. If any of these processes produce nitrosamine or contain precursor amine, solvents sent for recovery are at risks. So that when they are commingled, the introduction of nitrosamine impurities is possible. Recovery of the raw materials is often outsourced to a third party, so outsourcing can pose a risk. And if adequate cleaning of equipment is not carried out or it's not validated. The next thing is the quenching process. So when a quenching step is performed directly in the main reaction, that is when nitrous acid is added to a reaction mixture to decompose the residual acid. So this allows the nitrous acid to come in contact with the residual amines of the raw materials, and nitrosamine impurities could be carried to the subsequent step if they are not removed adequately. Even if the quenching is carried out outside the main reaction mixture, there is a risk of contamination if the recovered materials are introduced into the process. And the last is the lack of process optimization and control. So the lack of process optimization when reaction conditions such as temperature, pH or sequence of adding reagents are poorly controlled. Reaction conditions varied widely between batches and different processing equipment in the same facility. And the typical routine tests like HPLC for API purity, identity and known impurity are unlikely to detect nitrosamine impurities. and each failure mode could result in different nitrosamine in different amounts across batches from the same process and same API, with contamination detected if some batches but not all. So for the sources other than the APIs, nitrosamine impurities have been reported in many excipients in the ppm level. The nitrite impurities are found in range of commonly used excipients, which may lead to nitrosamine impurity formation during manufacturing and the storage. Supply qualification program should take into account that nitrosamine impurities vary across the excipient lots and may vary by the supplier. They are also found in potable water, and some drugs may undergo degradation pathways that form nitrosamine impurities that could potentially occur during the product storage. Guidance for industry gives recommendations. So manufacturers should consider potential causes of nitrosamine formation and evaluate risk for nitrosamine contamination in the APIs and the products. They should prioritize the evaluation based on the factors such as maximum daily dose, duration of treatment, therapeutic indication, and number of patients treated. So there are defined acceptable intake limits as per the guidance. They have given recommendations to the API manufacturers, and they have given recommendation to the drug product manufacturers, as we have seen in the previous slide. So it is a three-step process where the step one is to assess the risk of nitrosamine impurities in the APIs, marketed products, and the pending application. The step two is to conduct the confirmatory testing when there is a risk of presence of nitrosamine impurities. So due to some physicochemical properties, analytical methods for nitrosamine need to have a specificity, excellent chromatographic separation, and high sensitivity. And the step three is reporting changes implemented to prevent the reduction of nitrosamine impurities. So this includes the submission of any drug master file amendments or changes to the approved application. So the guidance and the recommendation provide the acceptable intake limits, and the acceptable intake limit is provided in nanogram per day for all these seven nitrosamine impurities into the USFDA guidance. If we can see what is an acceptable intake limit, so it is increased cancer risk of one case in 100,000, or in Indian terminology 1 lakh cases people, based on assumption of daily exposure to the impurity over a lifetime of 70 years. So if a person takes the impurity at in the medication daily at the lifetime for a 70 years, so there is an increased risk of cancer in one patient in 100,000 or 1 lakh cases, right? Now, limits are applicable only if a drug product contains a single nitrosamine of the above 7. If more than one nitrosamine impurity is present, the total quantity should not exceed 26.5 nanogram per day. If maximum daily dose is less than 80 milligram per day, the total nitrosamine of 0.03 ppm should not be more than 26.5 nanogram per day. And if it is more than 80 milligram per day, maximum daily dose, the limit should be adjusted not to exceed the recommended limit of 26.5 nanogram per day. If nitrosamine limits are without published limits are found, then ICH M7 should be used. Now in this case, further a recent revision of guidance, a new guidance on the nitrosamine drug substance related impurities have been defined, in which it has been defined on how to calculate the acceptable intake limit for other than these 7 nitrosamine impurities. Sensitive methods with LOQ in ppb should be used, and should be used with LOQ at or below 0.03 ppm. If more than one nitrosamine is detected, then analytical method should be validated for LOQs below 0.03 ppm to accurately quantify a total nitrosamine level of not exceeding more than 26.5 nanogram per day. Guidance make recommendation for the API manufacturers, firstly for mitigating the presence of nitrosamine impurities in the APIs. So it states that the manufacturer should optimize the design of the manufacturing process during route of synthesis itself to minimize formation of the nitrosamine impurities. They should avoid reaction condition that may produce nitrosamine, and if it is not possible, demonstrating that process is adequately controlled. Using bases other than amines, using a caution when route of synthesis involves the use of amide solvents, replacing nitrites with other quenching agents, optimizing and controlling the sequence of the reactions, the process, the reaction conditions like pH, temperature, etc. Designing a manufacturing process that facilitates the purging of nitrosamine impurities in the subsequent process steps. And they should consider the removing the quenching steps from the main reaction. When appropriate, they should establish control and consider additional specification for at-risk materials to prevent the contamination. Continuing with the mitigating the presence of nitrosamine impurities in the APIs, you should audit the supply chain and monitor them for any at-risk raw materials. The record should include the name of raw material manufacturer and supplier, role of the actual manufacturer, any repackers and distributors. To avoid cross-contamination, when recovered solvents are used, they should be used into the same step or in an earlier step. The recovered material should meet the appropriate standards before reuse. If recovery of material is outsourced to the third party, they should audit the contractors for the cleaning validation. Should also verify their suppliers whether the purchased material used in their process are recovered. To avoid unacceptable level of nitrosamine, they should analyze nitrite and nitrosamine levels in water and use water that has been purified. If nitrosamine is introduced to API through some exogenous source, then should eliminate the source of contamination. And API batches that may be reprocessed or reworked to control the level of impurities as per ICH Q7. If it is reprocessed or reworked in any way, the operation should be conducted oversight of the quality unit. Recommendation for the API manufacturer also includes the recommendation for control of nitrosamine impurities in the API. If a nitrosamine impurity is detected above LOQ, the manufacturer should develop a strategy to ensure that the level remains within the AI limit, and they should develop an appropriate control strategy including the specification limit. If the impurity is detected above LOQ, testing of each batch on release should be conducted. Alternate approaches like upstream test of an intermediate should be supported by adequate statistical control and should be submitted to FDA in a supplement prior to implementation. And any API batch found to contain levels of nitrosamine above AI should not be released in the market without prior approval of the FDA. Describe recommendation for the drug product manufacturers. The manufacturers must test representative samples of all the incoming materials, including the APIs at risk prior to use, and they should continue the testing until they have verified that API suppliers can consistently supply without unacceptable levels of nitrosamine. When designing the control strategy, they should evaluate whether nitrites could be present during manufacturing process where at-risk APIs are used. They should also evaluate whether nitrosamine could be formed in the finished product over the product's shelf life. If nitrosamine impurity is detected above LOQ, they should develop a strategy to control the level within the AI limit, which should include the specification limits as well. The control strategy is also recommended when introduction is inherent due to API structure, the ROS of the API or the manufacturing process. Testing of each batch on release should be conducted. Alternate approach should be supported by sufficient statistical control and should be prior approved by the FDA before implementation. If products which are already into the distribution have found unacceptable levels of nitrosamine, then they should contact the FDA so that they can be determined appropriate regulatory action. And any product batch found to contain levels above the AI limit should not be released for market and distribution. Maintaining the drug supply, the guidance states that if the manufacturing changes or recalls are likely to result into the disruption of the supply and a market shortage of the product, the manufacturers should contact the drug shortage department so that FDA can work with them to avoid interruption into the drug supply. And reporting changes to the FDA, it states that drug manufacturers must report changes implemented to prevent or reduce the nitrosamine in accordance with the regulations. If an API DMF holder makes changes to the ROS based on the risk assessment and the confirmatory testing, they must submit amendments and inform the manufacturer that references the DMF. If API is manufactured by the applicant but not covered by the DMF, manufacturers must report ROS changes in the application. If a batch of API is found to contain nitrosamine and is reprocessed or reworked, this should be reported into the DMF. If changes in the synthetic process is needed to avoid contamination, it is not possible to do it immediately. Manufacturers should submit both the process and the DMF with time frame for removal of the original process. Different synthetic process should be identified by separate codes. If changes to the drug product are needed to prevent nitrosamine formation, application holders must submit a supplement to FDA to notify any changes to the condition established. Holders of pending application must update their application through submission of an amendment. Provide the recommended timeline for completion of the three steps. So for approved and marketed drug products, step 1 risk assessment has to be completed by 31st March. So initial guidance was published in September 20. The FDA gave a 6-month timeline in September 20 to complete the risk assessment by 31st March. All the organizations have completed the risk assessment. The step 2 and step 3, that is confirmatory testing and submission of the required changes, has to be completed by 1st of October 2023. For the pending applications which are in the pre-submission stage, risk assessment and confirmatory testing has to be done prior to the submission, and if not available at the time of submission, may be submitted as an amendment to the original submission as soon as possible. And for the application pending with the agency, risk assessment and confirmatory testing has to be done expeditiously. Inform the FDA if the level is above the AI. If the level is within AI, however above LOQ, then amend the application accordingly. So this is all that is available into the guidance for the control of the nitrosamine impurities. Now we move to the next guidance, that is the limit, or how to determine the limit for the nitrosamine drug substance related impurities, which was published recently in August 2023. This is the most latest guidance that is published in August 2023, recommended acceptable intake limits for the nitrosamine drug substance related impurities. The content include the introduction, scope, background and recommendations for the acceptable intake limits, the implementation of recommended intake limits for the NDSRIs, and there's an appendix where provide the determination of the potency score. So at the beginning we have seen what are nitrosamine drug substance related impurities. They share a structural similarity to the API, having API or an API fragment into the chemical structure and are unique to the each API. So this guidance provides a recommended methodology for determining the acceptable intake limit determination using the structural features, right? So based on the structural features, a predicted carcinogenic potency categorization is determined and a corresponding AI limit has been determined for each of the NDSRIs. There's an uncertainty about presence and acceptable levels for these NDSRIs, and many of the firms are unnecessarily into panic and recalling it. So the guidance has been provided to ensure supply continuity of the drug and having an acceptable intake limit for the NDSRIs. It is applicable to all the approved or pending NDA, ANDA clinical development products and biologics as well. So it is based on the principles of ICH N7 that uses a structural activity relationship which is called as SAR concepts to assess and classify the mutagenic and carcinogenic potencies of the impurities. It considers that not all the nitrosamine drug substance related impurities have same carcinogenic potency as reflected by different recommended AI limits associated with each potency categorization. Mitigation strategy. So the mitigation strategy includes the supplier qualification considering potential nitrite impurities across excipient suppliers and excipient lots. Addition of antioxidants to the formulation that may inhibit the formation of the NDSRIs, and the excipients such as sodium bicarbonate that may modify the microenvironment to neutral or basic pH, which inhibits the formation of NDSRIs. Talked about the challenges in determining the acceptable intake limit for the NDSRIs, right? NDSRIs are unique for each API and therefore they are usually limited or no existing safety data, that is rodent carcinogenicity data. AI limit is available for some but not available for many of the NDSRIs. The compound specific AI limit can be calculated based on rodent carcinogenic data, that is TD50 values, that is published in some literature. TD50 values is available for approximately 140 nitrosamine, but they are mostly small molecules, and for other nitrosamines that is not available. However, many structures are not robust and cannot solely be relied on some calculating the AI limits. So the approach uses a SAR method to support identification of robustly tested surrogate, which is similar in structure and reactivity to the respective NDSRI, to generate an estimate of the carcinogenic potency, which an AI limit can be determined. It is important the rationale for choice of surrogate that is similar in activity and reactivity is very significant. So this method is known as the read across analysis. This guideline is based on categorization of the nitrosamine drug substance related impurities into different categories which according to their carcinogenic potency. Thus, it gives an introduction to the predicted carcinogenic potency categorization approach. FDA scientists in collaboration with other international drug regulatory authorities have developed this approach. This approach should not be used for the limits that are already defined for the nitrosamine impurities. This is a conservative approach representing the best available knowledge, and as the time and science develops, the limits may be changed. It is based on some activating and deactivating features like molecular structures associated with an increase or decrease in the carcinogenic potency. It assumes that alpha hydroxylation mechanism of metabolic activation is responsible for mutagenic and high potent carcinogenic response observed by many of the nitrosamines. This approach describes applies to the nitrosamine drug substance related impurities bearing a carbon atom on both sides of the nitroso group, where carbon is not directly double bonded to a heteroatom like N-nitrosamides, N-nitrosoureas, N-nitrosoguanidines and other related structures are excluded. The predicted carcinogenic potency categorization approach does not apply to the non-steroidal impurities where N-nitroso group is within an aromatic ring, that is indole. This guideline provides 5 potency categories rating from 1, 2, 3, 4, 5 and which provides a recommended acceptable intake for the nitrosamine drug substance related impurities in nanogram per day. For example, for the potency category 1, the limit is defined as 26.5, which is the limit for NDEA, which is the most potent nitrosamine impurity. The second is the recommended accepted impurity of 100 nanograms per day. So it is based on NDMA, which has a limit of 96.5 and hence this limit has given to be 100. The category 3 has a limit of 400 nanograms per day, which have a lower carcinogenic potency. The category 4 has a limit of 1500 nanograms per day. These are metabolically activated through an alpha hydroxylation pathway but are predicted to have a low carcinogenic potency. And category 5 also has the limit of 1500 nanograms per day because they are predicted to be metabolically evaluated as alpha hydroxylation pathway due to steric hindrance or absence of alpha nitrogen. Now we know that there are 5 potency categories, but how to put that nitrosamine drug substance related impurities into that particular 5 categories. So the guidance provide the determining predicted carcinogenic potency categorization and the associated recommended AI limits. So there is a decision tree kind of a structure given. So this applies to the NDSRIs bearing a carbon atom on both sides of the nitroso group where carbon is not directly double bonded to a heteroatom. For non-steroidal, for nitrosamine drug substance related impurities containing 2 nitroso group, the group with highest predicted carcinogenic potency category should be used to determine the AI limit. So this is the structure given where nitroso group where the definition of alpha carbon and beta carbon has been determined, right? So if we move to the left side of the decision tree, the first question is, does NDSRI have an alpha hydrogen on its alpha carbon? If the answer is no, the potency category is 5. So this is the table that we have seen. So if the question answer to the first question is no, the category comes out to be 5 where the limit is 1500 nanograms per day. But if the answer is yes, then it moves to the second question. Does NDSRI have more than one alpha hydrogen on one or both sides of the nitroso group? If the answer is no, the category again comes out to be 5 with 1500 nanogram per day limit. But if the answer is yes, it moves to the third question. Does NDSRI have a tertiary alpha carbon? If the answer is yes, the category is again 5 where 1500 nanogram per day is the AI limit. If the answer is no, then you have to calculate the potency score, right? So the potency score is calculated. We'll see how the potency score is calculated in the next slide. But if the potency score is greater than or equal to 4, so the question, if it is greater than or equal to 4, the potency category comes out to be 4. And if it is equal to 3, the category comes out to be 3. So for the value is 1500 nanogram and for category 3 it is 400 nanogram per day. If the score is 2, the limit is 100 nanogram per day, and if the potency score is less than or equal to 1, the limit is 26. That is the category 1, right? In the previous slide we have to determine the potency score. So how that potency score is determined? The potency score is alpha hydrogen score plus deactivating feature score plus the activating feature score. So what is the alpha hydrogen score? For example, one structure of a nitrosamine drug substance related impurity is present. So you have to count the hydrogen atoms on each alpha carbon, right? And you have to write it in a way that what is the least amount of alpha carbons, alpha hydrogens, and what is the highest amount of alpha hydrogens, right? So this table is specifically provided. If you can see example in the middle column, so if this kind of structure is present where on the left side of the alpha carbon there are 0 alpha hydrogens and on the right side of the alpha carbon there are 2 alpha hydrogens, right? So you on the left side you will write it in a way that 0 to 2, and for that the alpha hydrogen score is 3. You have to note that this is a standard structure, so you just have to note the structure given into the example, if that kind of structure is present and 0, 2 alpha hydrogens are present, the score is by default going to be 3. For example, in the second case, you can see on the left alpha carbon there are no alpha hydrogens, so the score 0 is written first, and on the right side there are 3 alpha hydrogens, so the score is written as 3. In that case, the alpha hydrogen score comes out to be at 2. So whenever you have an example where there are 0, 3 alpha hydrogens on the either side, that is 0 to 3 type of alpha hydrogen, then the hydrogen score is 2. Right. Similarly, other examples are also given where 1 and 2 alpha hydrogens are present, so the score is 3. 1 and 3 alpha hydrogens are present, the score is 3. And 2 and 2 alpha hydrogen is present, the alpha hydrogen score is 1. Right. Now the list of activating features and the scores. To calculate the activating feature score, some individual scores of all the listed features present on the NDSRIs. So there are two examples of activating features given. So it is only these two structures will have an activating example onto the nitrosamine drug substance related impurity. The first one is the aryl group bounded to the alpha carbon. So in that case, the individual score will be -1. And methyl group bounded to the beta carbon. So the structure example is given. In that case, the individual activating score will be -1. So similarly, a list of deactivating features have been provided, and whenever these deactivating features are present into the nitrosamine drug substance related impurities, a specific individual deactivating feature score is given. For example, if you can see the carboxylic acid group anywhere in the molecule will have a score of +3, and nitroso group in the pyridine ring will have a score of +3, etc. So during the determination of potency score, the team would have to determine the presence of these structures, that is either deactivating or activating structures, and if they are present in the molecule, they have to give the specific rating and consider that accordingly. This is given two specific examples for determining the potency score for better understanding. So the first example is for hypothetical NDSRI, that is N-nitroso 1-nebuvolol. The potency score, as we know, is the sum of alpha hydrogen score plus deactivating feature score plus activating feature score, right? So for example, if the structure is in this way, if we calculate the alpha hydrogen score, we can note that on the left alpha carbon there are 2 alpha hydrogens. On the right alpha carbon there are 2 alpha hydrogens. So we write it in a way as 2:2, right? So in this case, the alpha hydrogen score corresponding comes out to be as 1, right? So we have determined the alpha hydrogen score in that stage 1 as 1. Thing is the deactivating feature score. So from this structure that has been provided, the team determines from the list available into the guidance what deactivating feature is present, and if they find out this deactivating feature is present, that is hydroxyl group bonded to the beta carbons, the individual deactivating feature score will be +2, right? So the individual deactivating feature score becomes to be as +2. Now the team continues to look for the deactivating feature, individual deactivating feature 1, that is score we have determined. The other deactivating feature, if any, is present, and compares it with the standard list provided, and founds one another deactivating feature, that is chain of more than 5 consecutive non-alpha hydrogen items. So the score is coming out to be as 1, right? So deactivating feature score comes out to for any activating feature into the structure. So the team compares the activating any of the availability of any of the two activating features and do not find any of the activating features. So the activating feature score comes out to be as 0, right? So the potency score now we know is the alpha hydrogen score, which we have determined in the first slide as 1. The deactivating feature score, there were 2 deactivating features. So for the first one the score was 2 and the second one the score was 1, and the activating feature score, that is 0. So if we can sum all these 1 plus 2 plus 1, so the potency score comes out to be as 4. Based on the potency score of the molecule that is available, we go to our decision tree and notes that the last step, that is highlighted as red, calculate the potency score. So we have calculated the potency score, and if the score is greater than or equal to 4, the potency category comes out to be as 4, right? So in this case, we have determined the potency score as 4. So our acceptable intake limit is 1500 nanograms per day for that particular molecule. The second example for determining predicted score is N-nitrosoketamine. So this example, in this there is tertiary alpha carbon and there are alpha hydrogens, right? So we go to our questionnaires determining NDSRIs alpha hydrogen. So the answer is yes. the NDSRIs have more than one alpha hydrogen? The answer is again yes. Does the NDSRI have a tertiary alpha carbon? The answer is yes. Hence, the potency category is 5, that is the acceptable intake limit score is 1500 nanograms per day. In this way we have to calculate the categories, right? So if whatever the flow chart is given, if that comes out to be directly as category 5 or 4 from the questions, then accordingly the category is defined, and if not, then we have to calculate the potency score as we have seen in the example 1, and based on the value obtained into the category potency category score, we have to categorize into category 1 2 3 4 5, which will have a corresponding acceptable intake limit for that respective nitrosamine drug substance related impurity. Recommendation for manufacturers whose drug product contain multiple nitrosamines. So the recommendations made in this AI limits are for the NDSRIs, which corresponds to single NDSRI into a drug product. If more than one nitrosamine NDSRI is present, the total level of nitrosamine should not exceed the recommended AI for the most potent nitrosamine. Manufacturers may contact the agency if they are proposing any other concept apart from the most potent AI limit. So recommended AI limits for NDSRIs at risk for formation in certain APIs. Number of APIs have secondary or tertiary amines and are at risk of forming NDSRIs. Based on scientific analysis of chemical structure, it is concluded that when an API contains hypothetical nitrosated forms of secondary amine and dimethyl tertiary amine groups, recommended AI limits should be applied to NDSRIs to extent that they form in these APIs. So recommended timeline for the approved and marketed drug products. This guideline also defines the 3-stage process. Stage 1, that is step 1 is risk assessment, step 2 is confirmatory testing, and step 3 is changes to the manufacturing process and notifying the agency. So step 1 risk assessment has to be completed by 1st November 2023 for all the drug product formulation for the nitrosamine drug substance related impurities. That is within 3 months of the publication of the guidance in August. The step 2 and 3, that is confirmatory testing and submission of the required changes, has to be completed by 1st August 2025, that is 2 years from publication of the guidance. The implementation timeline includes the investigation of the root causes, identifying the effective changes into the manufacturing process, component supplier or product formulation, and confirming that any proposed changes will minimize the presence of NDSRIs. The firm must submit proposed formulation change through supplements or amendments to their applications. If NDSRIs fall at or below the FDA recommended AI limit, manufacturers should still develop an appropriate control strategy to ensure that the nitrosamine level is reliably at or below that AI limit. Right. So recommended timeline for the drug products in development or under FDA approval. So which are under pre-submission stage, the risk assessment and confirmatory testing has to be done prior to the submission. If it is not available, may be submitted as an independent amendment to the original submission, and amendment should be submitted as quickly as possible. So which are pending with the agency, the application, the risk assessment and confirmatory testing has to be done expeditiously, and inform the FDA if level is above the acceptable intake limit. Recommendation for the marketed products with NDSRIs above FDA recommended AI limits. So they should not be released by product manufacturers for distribution, and they may warrant a removal from the market. If the marketed products contain the levels above the AI limit and firm is proposing a recall which might disrupt the supply, they should contact the FDA drug shortage department. FDA may work directly with the specific manufacturer and should consider whether it is appropriate or not appropriate to recommend an interim AI limit for a temporary period. Approaches to justify a proposed alternative AI approach. So should submit scientifically justified rationale associated with predicted carcinogenic potency category. Alternative approaches using safety data, that is either obtaining a compound specific data or using a read across assessment to a suitable surrogate. So the mutagenic assessment, when testing NDSRI using bacterial mutagenicity assay, use of full complement testing strain described in the OECD utilizing pre-incubation method and both rat and hamster S9 at a concentration of 30%. Recommends using S9 fractions typically prepared from animals treated with an inducer of CYP450 like a contribution of phenobarbital and beta-naphthalene flavone. Manufacturers and applicants should use recommended pre-incubation time of 30 minutes as part of optimal condition for detecting a signal for mutagenicity. And the read across assessment based on the surrogate, so can perform QSAR analysis to predict mutagenic potential by following recommendations that are described in ICH M7. Selection of surrogates with robust carcinogenicity data. Robustly tested surrogate nitrosamine is frequently unavailable. When justified, following compounds should be considered like NDMA, and different examples have been provided here. We come to the end of this presentation. Hope you have liked the presentation. Kindly subscribe to the channel Prof Talks for more such informative videos.